Excellent. Thank you so much, Jenny, for that detailed walkthrough. We'll move straight into the second LANL presentation. So now I'd like to introduce Dr. Jennifer Mamrosh to present the LANL HIV Immunology database. Thanks, Jennifer.
So we will talk today about why the HIV immunology database is important. So we really need to have better understanding of HIV immune responses if we ever want to develop a vaccine for it. And this would be really transformative, especially in poorer countries. And for most approved vaccines, I mean, vaccines have been around a long time. Often the first strain that anyone ever tried to inject into a person of a virus or bacteria worked. People develop beautiful antibodies. And so these are, typically very minimally engineered immunogens. You often get 1 to 3 administrations of the same thing. But that clearly isn't going to work for HIV. It's been tried for decades. As of right now, I mean, the HIV vaccine efforts actually look really promising, but most likely what will have to happen is you'll have to have a highly engineered immunogen that was designed to try to engage rare B cell populations. So we have to really understand HIV biology to design these. And then most likely people will have to be injected with multiple different immunogens at different times so that their antibodies can be shepherded down these improbable paths to develop the mutations required to actually have a lot of breadth. And this is, completely different from any other vaccine strategy that I know of for a different pathogen. And this is really because of HIV's high mutational rate. So it mutates, far faster than human DNA, about 2,000 times faster than COVID-19, and about 200 times faster than influenza. And we all know that the influenza vaccine is often difficult to design and not super effective against prevailing strains that emerge. HIV has some additional challenges. The envelope protein, the main antibody target, is heavily glycosylated. There's also latent HIV reservoirs in people's bodies. And ultimately, what you can take away from this slide is that immune escape is just so much more pronounced in HIV versus nearly any other disease. So the immune response to HIV consists of both T cell responses and antibody responses. So here on the right shows two different types of T cell responses. You have CD8 positive T cell responses, cytotoxic T lymphocyte driven. These are MHC class 1 dependent. The peptides presented are quite short. And CD4 positive T cell responses are class 2 dependent. The peptides are a little bit longer. We don't know as much about CD4 T cell responses in HIV. I think that they are actually quite important. They're harder to study. But both the T cell responses appear to occur quite quickly in response to viral infection. And antibody responses take a little bit longer to develop. The epitope for antibodies can be long or short. It can be discontinuous. And the best antibodies for HIV take years to appear. And only a small percentage of people clear HIV infection on their own. Only about 1% of individuals have good enough T cell responses that they can actually eliminate the virus. And, somewhere in the range of 1 to 10% of individuals make good enough antibodies to control HIV. So, this is unlike almost any other virus where you get infected, but nearly everybody recovers and develops immunity without additional treatment. So T cell responses to HIV are often quite effective initially. So here's two different individuals from this publication. If you look at the first couple of years, individuals' T cells are targeting this epitope here in blue, and the T cell response is quite effective. But over time, this epitope starts developing mutations, shown here in red, and T cell responses to this mutated epitope become less and less good. So the person is losing control of the virus over time, and eventually they'll progress on to AIDS if they're not treated. So HIV within hosts actually evolves really similar to how cancer evolves. You get selection for an expansion of HIV clones that contain beneficial mutations, typically those facilitating immune escape. And And antibody responses to HIV, like T-cell responses, initially are actually pretty effective. But the thing is that even though you can develop a good antibody response to the infecting strain of HIV, that antibody is typically not effective anymore once HIV evolves. It takes a long time for what are termed broadly neutralizing antibodies to develop in a small percentage of individuals. It typically takes maybe 2 to 3 years, and this is because the antibody has to co-evolve alongside the virus to develop breadth against many different viruses, viral sequences. So if you're interested in HIV immune evasion, our database has since 1994 curated information on immune responses in what we call the HIV Immunology database. And together with the sequence database, we were the first NIH-funded host pathogen databases. So you can get to our databases using this link. And as Jenny mentioned, we have several different databases on our website. We try to connect them as much as possible. The immunology database has information mostly on antibodies and T-cell epitopes. And it sometimes has information when it's available in publications on the individual from which the antibody or T-cell response was characterized. And when possible, we link it to sequences of HIV in the HIV sequence database. There are many tools that work with immunology data. We connect the immunology database very often with data in the neutralization database, the CatNap tool, and antibody neutralization data. And we also connect the immunology database with what we call the antibody context database. And this will tell you the activity of antibodies for different regions of the envelope protein. So our data curation process, like a lot of the other databases for our site, consists of both manual and automated identification of publications. So we're only looking for select data. So we're looking for immunogenic T cell, both CD4 and CD8, epitopes against HIV-1 and HIV-2. Occasionally we curate something else. We don't do that much. We don't take data for non-immunogenic epitopes except for variants of known immunogenic epitopes. So unlike IEDB, we don't put in a lot of data for things that have been reported in a publication to not be immunogenic at all. And we also curate data on antibodies against HIV-1 and HIV-2. And then we have our human annotators like IEDB, PhD level scientists that triage the publications. We find the ones that will have the biggest impact to the HIV research community to include on our database. And we manually curate the data from publications. So a lot of the data that we take for our database is similar to what IEDB takes. We definitely want, of course, the T-cell epitope sequence. We want associated data, like what was the MHC or HLA allele, viral subtype. We take antibodies. We collect information on, what was the immunogen? Was it infection? Was it vaccine? What was the species? Something that we do that IEDB doesn't is we'll take information for antibodies that have no known epitope. And this is, I think, sort of also due to the history of our database. We were started, a couple of decades ago, and there was just less of this information available often. We try to collect information on the patient or individual from which the immune response was characterized. And we have our human annotators write a summary of publication findings for each of our records. So they're not summarizing the whole publication. They're summarizing for that particular antibody or T-cell epitope what the most important findings from the publication was. So if you read these summaries, it's actually a really informative way to get information from the publication without having to read the entire thing yourself. And at this stage, we often have substantial discussions with publication authors. And we do a yearly review of our records by another individual than the one that actually curated the information. And we're always considering updating or adding new types of information to our records. We often go back, 20 years and do quality control projects on older records where we're going back to the publication and adding additional things that we didn't curate at the time. So today, I'm going to do 2 research examples with some live demonstrations to describe how to use our immunology database. So this is not going to cover everything that you could do on the database or everything that's available. I would encourage you, if you're interested in something, we don't cover it, to go to our website, see if there's a tutorial that covers this additional topic. And the slides from today will also be, they're actually already available on our website if you want to look at them more closely. So the first example looks at mutations in the HIV GAG protein and HIV long-term non-progressors. So this is going to focus on CD8 positive T cell responses. So here's just kind of a summary of, just basic information about these. So the study looked at people that are termed HIV long-term non-progressors. They looked at 19 individuals, and these are people that were actually not being treated with antiretroviral therapy, but yet their disease did not progress. At least it did not progress for some period of time. All of these individuals carried what's considered a protective MHC class 1 allele, which is HLA-B27. The HIV GAG protein was sequenced at the study start and last follow-up, and unfortunately, during the course of the study, some of the individuals did start to have their HIV progress. So the investigators of the study sequenced the GAG protein, and they saw that certain mutations were identified in it, and some of them appeared over time. And I mean, that's not unexpected because HIV mutates quickly. And here's just a short region of the GAG protein I'm showing here for simplicity. But they asked the logical question: do any of these mutations that are appearing over time actually directly contribute to disease progression in certain individuals? So we will consider just one mutation for simplicity today. It's GAG amino acid R264. And in particular, we're interested in when it mutates to a lysine. So say you had just a sequence or a much longer sequence, and something interesting seems to be going on. You think escape mutations are appearing in it, but you don't actually know. I mean, is this a T cell epitope? Is it an antibody epitope? I mean, what is the immunological significance of this? So how can we know where to even start looking into what the effect of the mutation might be? So one way is to use our HIV genome browser tool. So this is probably like the most intuitive and interactive way. So I'll do a quick live demo of that. So if you go to HIV genome browser, you can select the GAG protein, and over here, let me just get rid of this. You can pick different tracks. So you can pick, say, antibody linear epitopes. And we can go to the region of GAG that we're interested in, like around 264, and zoom into that region. So that would be this amino acid right here. And you can see that there's a couple of antibody epitopes spanning this region. If you go down and you look at CTL epitopes, you can see that there are many, many more. If you scroll down, you could see that there are many T cell epitopes within this region of the GAG protein. And if you're really interested right away in like understanding the immunogenicity of epitopes, we also have a track for that. You can look at epitopes like this one here spanning that amino acid is immunogenic. All the ones that are in green are immunogenic. And if you go down, you can find some examples that, probably have escape mutations. Like this one has reduced immunogenicity. This one is no longer immunogenic. So by looking at that, I mean, it really appears that if you had to pick something just based on the number of things that have already been found about that region of the GAG protein, it's probably a CD8 CTL epitope. Another way to look for T cell epitopes is to use our epitope location finder tool. You can put in a pretty long sequence. Here I put in this sequence. It says that this exact sequence is an epitope. I mean, okay, it's kind of a cheater example, but we tried to make it relatively straightforward for today. So it comes up as an epitope. The HLA restriction is B27, just like the individuals in our study. If you were to click on the link, it will bring you to our database records for it. And I also found, some other epitopes that have, say, mutations in them or restricted by other HLAs. So it gives you kind of a summary as to what is known for epitopes within that sequence in our database. And another thing that you can do is to actually do searches for epitopes. So we'll go to our CTL epitope search and do a live demo. So we go to the immunology database, and the link is up here. So I'll pick the GAG protein, I'll pick the epitope, I'll say exactly match just so that we get less results. I'll pick the restricting HLA of B27, and I won't pick anything else. But if you want to say like you only care about ELISpot assays for interferon gamma, you can pick that, or you can pick the immunogen being infection, or we have many options. So let's hit search, and what comes up is 43 matching records. So each of these records comes from a separate publication. So this publication, they looked at individuals. The HLA restriction was B27. They did a few different experimental methods to characterize immune responses. And the notes from the publication are here. So if you read this bottom one, it says that this epitope was recognized by 3 out of 5 HLA B27 positive patients. So there's already some evidence that this is indeed immunogenic. And there's more, detailed information available above. And if you want to know, like, is this epitope likely to actually be found in HIV subtypes around the world? If you click on this link, it will show you the frequency of the amino acids in this epitope and all other amino acids in the region that this is derived from in the GAG protein and different HIV sequences. So you can really get a sense as to like the likelihood of escape mutations in different populations. So then you can ask the question, okay, so we know the epitope that's non-mutated is immunogenic. Like, does an R264 mutation render it no longer immunogenic? You'll incidentally find some of this information if you're looking through our CTL epitope search results, but we have a separate interface, which is called the variant search interface, that's more specifically set up to address these kinds of questions. So we will go there. So it's here, it says epitope variant search. And we'll select similar things again. I'll pick exactly match. I'll pick the HLA allele and hit search. So what comes up for variants of the immunogenic records is 8 matching records. So this is one publication here, and there's summaries of the publication findings here. And then there's information for the specific variants here. So this is the one that we're interested in. It's an R264K mutation. And if you click on it, you can expand it and get some additional information. Here, the mutation type says calculated escape. That means that the variant was found more often in HLA matched than HLA unmatched participants. So, I mean, there's some statistical evidence suggesting escape, but there's no actual evidence for immunogenicity. So if you're looking for things like that, I would encourage you just to keep exploring the results. The second result here, if you click on this epitope that we're interested in with the escape mutation, it says that this was, previously documented as an escape mutation, and when the individuals did ELISpot assays on it in B27 HLA expressing individuals, they did not see an immunological response. So I would say in summary, there's ample evidence from multiple publications that mutations at R264 can confer immune escape in individuals with this HLA allele. And hopefully this shows our database can be used to obtain publication data for HIV immunogenic epitopes, even if you just have a longer sequence and you don't know where the epitopes are, or if it's an antibody or T-cell epitope. We have a lot of different ways that you can actually figure out what the immunological determinants are likely to be within your sequence. So this is just a cheat sheet. It's available on the slides on our website. So let's move on to the second example. So this is understanding escape of the envelope protein from broadly neutralizing antibody pressure. So, again, in response to HIV infection, just a small percentage of individuals, probably about 2%, will develop, and it takes a couple of years, broadly neutralizing antibodies. And you can isolate these from people. Now, several different ones have been given to other individuals therapeutically to control or prevent HIV infection. And a lot of times they work for some period of time, maybe it's a couple of years, but eventually HIV will typically mutate away from the antibody pressure. And for HIV, I mean, antibody epitopes are often discontinuous, just like they are for any other pathogen. And they're often against very specific regions of the envelope protein. And this is mostly due to how much of the trimer is actually glycan shielded. So antibodies kind of target, just a few sites that don't have glycans on them. And our database contains exact epitopes for actually only a small number of antibodies. Unlike IEDB, I mean, we're willing to take data for antibodies where we might say like the epitope is somewhere within the CD4 binding site. We don't actually know what it is. I mean, we know the coordinates of this binding site, but we don't know what the actual contact sites of the antibody are. But, from our perspective, that's okay. We take that kind of data. So the study that we're going to look at today wanted to understand how envelope protein mutations confiscate from neutralizing antibodies. And they did what's called deep mutational scanning. They looked at libraries of HIV with single point mutations in the envelope protein. They incubated them with a specific antibody. They did selection. And then they looked at mutations allowing for viral escape. So we're only going to look at one antibody today, which is called 10-1074. And the authors of the study found that there are really only three main positions in the envelope protein that mutated in response to antibody selective pressure. So it's D325, N332, and S334. And if you don't know anything about the antibody, I mean, it's entirely unclear why these particular sites might be important. But we'll use this to show you how you can get more information for antibodies in our database. So you can actually do an antibody search in our database for this exact antibody. So if you go back to the main immunology page, click on antibody search. Again, there's a lot of options you could choose. You could choose like you only want neutralizing antibodies that are against subtype A HIV. But for today, we'll just look at that exact antibody since we already know what it is. And hit search. So what comes up is one record. And for antibodies, we actually have a lot of information available, often from multiple publications. So what's up here is a summary of information that's consistently true across all publications for the antibody. I mean, it might be like antibody contact sites, what the binding region is. This was an antibody elicited against subtype A HIV infection. And if you click on notes, we have 101. So this is from 101 publications. And these are all like human written notes summarizing specifically for that antibody what the most important findings were for the publication. So, this note kind of describes isolation of this antibody from an African donor. So you can read these notes to get, I think a pretty deep understanding of what's important to know about the antibody without having to go to the actual publication. And we have a lot of additional information available. If you're interested in like where the antibody is known to contact the envelope protein, we now offer a link to a 3D viewer. So we have the antibody binding sites shown here in red, and this is the envelope protein. And if you're interested in, say, a particular site, we'll pick N332. You can hit mark and we'll show you where it is. So this is actually within where the antibody is known to contact the envelope protein. So you can go back and we can click on some other links to get more information. If you go down here to where it says catnap data, this is going to the catnap neutralization database. We might be able to find the antibody sequence, neutralization data for it against different viruses. If you want more information on the individual from which the antibody came, you can click on this link if it's available. And then the contacts and features database, if you click on this, it will give you very specific information on different things related to how the antibody interacts with the envelope protein. So here are the contact sites of the antibody. And we were interested in 332, 325, and 334. And actually, all of these are represented here. So it's clear that, this antibody is causing mutation or enriching, selecting for mutation of the envelope protein at sites in which the antibody actually contacts it. I mean, that, seems intuitive. If you want additional details, you can click on this link and go down. There's, very specific information from the publication. It will tell you that the 332 amino acid in the envelope protein is a common glycosite. So, in summary, we have a lot of information on our site for 101074. That's not true for every antibody, but this is a highly studied one. The envelope protein is probably mutating away from antibody selective pressure because it's the sites that the antibody is known to contact the envelope protein are what are mutating in response to pressure. So I think, that's not surprising. Say you were interested in finding antibodies targeting similar residues to 1074. What you could do is go to the search interface for the contacts and features database. If you put in 332 here and you can select contacts and hit submit, you'll see that about a dozen other antibodies also can contact that glycosite. So say you wanted to find antibodies that don't target that residue. And this is actually a question that will become increasingly relevant as people receive monoclonal antibodies more as therapeutics. In wealthier countries, a lot of people with HIV are now in clinical trials in which they're being given monoclonal antibodies to either prevent or try to treat HIV infection. And these antibodies often work for some period of time, but eventually people develop escape. So say, you're a clinician and you're treating somebody with 101074 and it worked for several years, and then all of a sudden their disease starts to progress and you wanted to find a different antibody to use. You could do crude things like saying, okay, 101074 targets like the V3 region and glycans of HIV. I'll just pick a totally different region like this EMPER region and find antibodies that target that, and it probably won't depend on the same contact sites. I mean, that probably will work. But if you want to do this in a better way, that would be using our CatNap tool. So as Jenni talked about, this aligns viral sequences with antibody neutralization assay values. And you could use this tool to find antibodies that have neutralizing activity, even if you have viruses with mutations at these amino acids. So I will do a quick demo of CatNap today. So there are many antibodies. We have over 1,000 that you can select. I'm going to pick just a few of the most commonly studied ones for the purpose of this demo, including 101074. And then we have almost 3,000 viruses you can select. That's too many to look at right now. So I'm going to pick this global panel of viruses that only has 12. And I'll hit submit. So this brings you to this page. And I mean, I'll admit it looks a little bit complicated, but it can actually be used to do pretty powerful analyses. So on the left here are the virus names, and then the virus sequences here on the right, and you can scroll through them and see differences between them. And then in the middle here is the neutralization assay data for different antibodies. So this is 101074. This is another antibody. This is another antibody. So they all have their own values. And the values that are in red or orange means that the antibody is very neutralizing, which is good. I mean, that's like antiviral activity. And if you see UD or values in green, I mean, the antibody is a very weak neutralizer. You want to use it in any kind of therapeutic context. So if you look here, you can see that this antibody is very effective against certain virus strains, and it's not effective at all against others. And if you wanted to try to understand why that would be the case, what you could do is you can sort the values from most neutralizing to least neutralizing, and then you can start looking for sequence features that are associated with that. And we'll go to the 332 glycosite, and you can see that here, all the antibodies that are neutralizing, or I mean, all the viruses that are neutralized by 101074 have a glycosylation site at this residue, and those that are not neutralized don't. And this is probably not enough to do a statistical analysis. You could analyze more viruses, but it will try to give you a sense as to what the important sequence residues are for virus neutralizing for antibody neutralizing activity. And say you want to find an antibody that would work even when 10-1074 doesn't. I mean, I would say that like looking at these values, this one is very neutralizing. It clearly is neutralizing even if you don't have a glycan or a potential glycosylation site at this residue. So if someone developed resistance to this antibody, you could potentially give them this one. I mean, you would want to look at other sites that you knew escape mutations accumulated at too. But the CatNap tool can be used to do these kinds of analyses. And you can actually do significance analyses of specific amino acid positions within the CatNap tool. Like if you wanted to test, is it statistically significant that viruses neutralized by 10-1074 antibody have a glycosylation site at this position? You can do that type of analysis actually within the tool. So again, here's a cheat sheet of how to search for antibodies. You can find it on our website in our tutorial section. I'll cover very briefly just a couple of tools that we have. A lot of our tools can be used for any species. So the ones in blue here, if you have custom input, you can use the tool. So you can predict optimal neutralizing combinations of up to 4 antibodies. We have 2 tools that are HIV specific. One of them that are listed on this slide. One of them predicts the location of glycan holes within an HIV sequence. We have a tool that's much more general purpose. I think a lot of people use it for like non-viral pathogens. I mean, you can just predict N-enolene glycosylation sites within a sequence. We have a tool that's pretty widely used to develop T-cell epitope vaccine antigens, and these are ones that should have optimal population coverage. We have an HIV-specific tool called Cabin that allows you to search for antibodies that have specific neutralization properties. I mean, it's different from the CatNap tool. I would say is complementary. And we have a lot of MHC, HLA associated tools. You can find potential HLA enriched epitopes across sets of individuals. You can compare allele frequencies across populations, or determine if these are significantly different between 2 populations. And we have a recent tool called the LANL HIV Immunology browser. So this is an interactive browser in which you can look at immunogens. So these are HIV vaccine immunogens. They're based on the envelope protein. They all have different names and different sequences. And if you hover over specific mutations made in it versus the parental sequence, it will tell you up here the consequence, why this mutation was introduced, how it's beneficial. So lastly, we have a couple upcoming improvements. We don't have a firm release date, but we're hoping within the next year that we're going to release an antibody paratope viewer. So this would be specific for HIV antibodies. It would be interactive. It probably will look a lot like the LANL HIV Immunology browser. And then we also plan to add T-cell receptors for known immunogenic HIV epitopes. So lastly, thank you so much for attending. We're really funded to help you with your research. We're happy to be contacted with training requests. We often work with people one-on-one to help them better use our database. A lot of our tools have actually come from user suggestions. So if you have any ideas for interesting tools, even if they're not entirely HIV specific, please let us know. And we do a lot of collaboration with academic and external researchers as well. We're happy to do that. So you can contact us at either of these email addresses. So thank you so much. I think I'll stop sharing and we can go to the Q&A.
Thank you so much, Jennifer, for that great presentation and the live demonstrations as well. That was awesome. We do have a few questions coming through, so I'll give you a minute to take a look as well. And I'll invite Jenny back to the virtual stage. And Jenny, if you would like to take the first question.
Sure. So the first question is from anonymous attendee. Is the download only available by user interface? Do you have APIs? So I believe this question came in while I was talking about the sequence database, but I'm going to take a little bit broader perspective on all of our databases. Most of our databases do have full database downloads. I think we are just about to make those public for the immunology databases. We definitely have full database downloads for our feature database and for the catnap neutralization database for sure. Unfortunately, we have not been able to set up an API for the sequence database at this time. We have some technical hurdles that we haven't been able to get past. We do, however, have bulk download instructions. So if you go to our main search interface and look for those bulk download instructions, they will kind of walk you through how to use our advanced search interface to download. You can, in fact, get all of our sequences if that's what you want. In most cases, if we correspond with an author and ask them, what they want to do, it often turns out that our curated alignments serve their purpose, or that they can download a subset of our database. They don't really need every single sequence there. A lot of it is junk, to be honest, since we're taking everything out of GenBank that's HIV. So I would, if you want to do a bulk download or something like that, you're welcome to contact us. We'd be happy to help.
Okay, so I'll take the next one. So the question is, can the data be downloaded for multiple peptides? And if yes, do you have a limit? Simplifying the question a little bit. And can we download data using APIs? So now, as Jenny mentioned, for the sequence database, we don't have a way to download everything. We offer some suggestions on our website for how you can download in several steps the entirety of the sequence database. For the immunology database, now you can download all of the information. You can't do it all in one place. So if you are interested in, say, CD8 positive CTL epitopes, if you go to the CTL epitope search interface near the top of the page, now there is a link to download either a CSV or a JSON file. So if you click on that, you'll get all of the peptides, the CTL peptides in our database. We regenerate the files weekly, so it's possible it will miss a couple, but you'll get all of them. So we don't have any limit on how many you could get. And that's a pretty new addition that we've added. We do also have an API for the immunology database, but I would say it's probably simpler to just download the JSON or CSV file and filter it as you would like. And if people ever have questions like they can't figure out how to find these files, or you need other data that's not contained within them, I think pretty much everything is, but we can try to work with you to help you get the data that you're specifically interested in.
Okay, are we ready for the next question?
Mm-hmm. Yeah, go ahead.
Anonymous attendee asks, are all tools user interface based? So to regenerate the same results, is there a way to track back the selected options? So most of our tools are user interface based. They can accept fairly large input. We are aware that people do request from us sometimes the scripts so that they can run our tools locally. And we are working on that. Some of them are available. We have some hurdles because we work at a national lab, which doesn't like us to release scripts without certain levels of permission. But we are working on making more of them available so that users can run them locally. We don't have any way to regenerate the same results, like to track back the selections, unless the user keeps track of that themselves. I guess our philosophy on tools is to make things as widely accessible and user-friendly as possible. But so one thing, we don't require a login. So in order to track back, particular selections and redo an exact search, you'd probably need a login, and we don't require that.
Yeah, just one like quick thing to add. I mean, if anyone's ever in a desperate situation, like you ran, say, catnap 2 years ago and the results were really interesting and you put it into a publication, you submitted it for review, and the reviewer said, like, what exactly did you select? And you can't remember. And catnap doesn't work the same way now because we've added a lot more data to it, or we've changed the scripts. I mean, it fundamentally works the same way, but just like some theoretical example like that. If you contacted our group, I mean, we're willing to work with people. I mean, we can go back and look at changes we made to the code. We can go back and look at, what data was actually in the database, say 2 years ago. We're not going to analyze any of the newer data. I mean, it's, it can be some work, but if you're ever really desperate, I mean, if you ran something in the past and you just can't replicate the results anymore because data in our database has evolved and we've added more, I mean, come and contact us because we often can help you in that kind of situation. So then I will answer another question. How would I search for NIH HIV predicted T cell epitopes that are being targeted by specific HLA alleles? So I would say we don't have what I would consider predicted T cell epitopes. Our database only curates information on epitopes that have been experimentally tested, except for variants of known immunogenic epitopes, but we don't have like a region of our database in which you can look for, like, say, for a specific sequence, what would be a predicted epitope. We have some tools where you could put a sequence in. We have like an H epitope tool in which you can put a sequence in. It doesn't even have to be HIV specific, I don't believe. And you can pick HLA restriction and it will tell you potential epitopes within that sequence. It works, I think, similar to non-MHC2 pan. It's not as sophisticated in terms of the analysis, or not MHC pan. It's not as sophisticated in terms of what it actually does computationally. But we have some tools for finding predicted epitopes. But we don't have predicted epitopes themselves stored in our database. But we have epitopes for specific HLA restrictions. So if you wanted to find known immunogenic epitopes, if you go to like our CTL CD8 positive epitope search, you can pick specific HLA alleles and find epitopes known to be restricted by those alleles that are immunogenic. So hopefully that's a little bit better than even predicted. But yeah, we don't include predicted ones.
I think the next one, you probably should take that one too, Jennifer.
Oh, sure. So, okay, have you done a benchmark between your epitope repository and HIV and IEDB? How much overlap is there? So there's intentionally relatively little overlap. We do share our data with IEDB so that they can put it on their website. I mean, this is something that is only recently happening, so they don't have much of our data. In the future, they will have more of it. It's not going to be continuously updated because we don't have a great process to do so. IEDB doesn't curate information on HIV immune epitopes or antibodies unless they're working on that paper for some other reason, say like they're interested in antibody that reacts to CMV and it has cross-reactivity to HIV. Stuff like that can make it into IEDB, but they don't really do their own curation of HIV immunological information. I mean, this is kind of just an agreement between our funders. We're both funded by the NIAID, and they said that there's no reason to duplicate effort across the two databases. And I think that's all for questions.
Excellent. Thank you both so much. Thank you, Jenny. Thank you, Jennifer, for those presentations and answering all the questions so thoroughly. And we got to thank you. Thanks to the anonymous attendee who just posted that as well. So if there are no other questions or comments, I'll thank you both again so much. And thank you to the LANL team for joining us. They've been joining us for multiple years now. And it's really, really nice to be able to showcase other resources outside of the immediate IEDB and CEDAR team as well. So thanks, Jenny, and thanks, Jennifer.
Thanks, Nina.
Thank you.